Click for IB Biology. We're now looking at the phenomena of surface area to volume ratios in organisms, and in particular at the cellular level. There's a PDF of this presentation down in the description, where you'll also find 10 questions with the answers, of course, so that you can check your progress. Surface area to volume ratios. So one of the first things that we discover as we intensify our study of biology is the variation in cell size that we can find. And perhaps we could start by looking at the idea of the diameter of the head of a sperm cell with its nucleus is approximately 5 micrometers, whereas the human egg cell has a diameter of something like 100 micrometers. So in diameter terms, the sperm cell is a 20th of the diameter of the egg cell. So a considerable size change there. Red blood cells have a diameter of about 7.5 micrometers, whereas a white cell would have a diameter of nearly 20 micrometers. Neurons, whilst their cell body is relatively small, the extensions of the cytoplasm in multiple directions, which is characteristic of a dendrite, mean that some of those cell lengths are 100 micrometers. So that is 1/10 of a millimeter. However, these cells are dwarfed by striated muscle cells, which can be measured in millimeters, or indeed some of them even in centimeters. These incredibly long cells, which we would find in the long muscles such as our quadriceps or our hamstrings, can have lengths of up to 30 centimeters and diameters of 100 micrometers. So incredibly narrow, a tenth of a millimeter, and yet 30 centimeters long. So tremendous ranges in size, and there are consequences to changing size. And we can summarize the consequence of changing size of a cell by saying that as we increase the size of a cell, what happens is that the surface area to volume ratio decreases. So let's take a look at what this actually means for the cell. And here we're using a model which is given in units of centimeters, but of course real cells would be in micrometers, but this is just as a mathematical model. So let's look at the small cell, which is over here. So with a 1 by 1 dimension, each surface has a surface area of 1 centimeters squared, but of course this is multiplied by 6 to give us 6 centimeters squared. And so the surface area is 6 centimeters squared, but the volume is 1 cubic centimeter. So we have a ratio of 6:1. So let's think of it like this. For this 1 unit of cell, so let's think of cells now, it has 6 surfaces into which diffusion, as an example, can occur. The 6 faces of the cube. Now let's jump ahead to this larger organism here, which is 3 by 3 by 3. And so the calculation then is that the total surface area is 54 and the volume is 27. And when we make that a ratio, we end up with a ratio of 2:1. So indeed, the surface area to volume ratio has decreased. But what does this actually mean for the organism which has got larger? Well, let's look at the cells on the surface, or the sections of the organism on the surface. If we take one of the units on the corner, this one would be better actually, we have 1, 2, 3 exposed surfaces for the 1 unit of volume. Unlike the original one, which had 6 surfaces. So this one has 3 surfaces for 1 unit of volume, whereas this one had 6 surfaces. So if we think in terms of diffusion, we can understand that now the larger organism is experiencing problems. But we're not finished, because if we take this cell here, it has 1, 2. So this has 2 surfaces for 1 cubic centimeter of volume. And of course, if we look at this cell here, then that only has 1 unit for 1 unit of volume. And so the surfaces are getting smaller and smaller. And of course, some of you will have realized that in the center of this cube, there is a unit of the organism which is in the center of this cube, which is not exposed to the exterior surface for diffusion at all. And so getting larger has created real problems for this organism in terms of diffusion of substances into and out of the cell. We say that the surface area to volume ratio has decreased with an increase in size. Just before we leave this point, we can actually express this in words where we can say that the surface area increases by the square of the length, but the volume increases by the cube. And this is what creates the compromised position for getting larger. So cells need to maintain diffusion, and one of the ways that they have around this issue is to modify their surfaces so that exchange rates by processes such as diffusion become more efficient. So they express genes that change their shape, they differentiate, and they specialize their shape, and they specialize their function. So here's 3 examples of that phenomenon. This is the first one, is folding. So the response to getting larger can be, I can fold the surface in. So we have here a red blood cell where the surface has been folded in on 2 sides in these circular cells, and this is known as a biconcave disc. So the surface area has been increased by folding in. And then here, this is a diagram of the proximal convoluted tubule of the kidney nephron, the first section of the kidney nephron. And this also has problems with surface area to volume ratios. And if you look carefully here, you'll see that as substances move through, the filtrate moves through this tube here, the cells have increased their surface area by folding out their cell membrane, and this increases the surface area for absorption. And here's an electron micrograph of cell membrane being folded in order to create these villi type structures, microvilli structures, to increase the surface area for absorption. So if you want to keep diffusion happening at a higher rate, either you're going to fold the membrane in, or we can fold the membrane out. But another strategy for rapid diffusion is to flatten the cellular structure. And this is very much seen in lung tissue, where the cells of the alveoli are greatly flattened in order to reduce the distance between the oxygen in the air here and the blood cells and the blood supply, which is in the tissue here. Here's a closer look at that image. So we're still with lung tissue, and here we find 2 types of cell. And the first one is called a pneumocyte type 1. And pneumocyte type 1s have very, very flattened structure. Notice that there's alveoli space here and here, and this is true for the entire structure. So there's an alveoli, and here's another one out here, but we've got oxygen here, and here's the alveoli. This is the pneumocyte type 1. So they have been flattened in order to reduce the diffusion distance. Also within these lung tissues, there's a second type of alveoli cell called a pneumocyte type 2. And what they do is that they produce these vesicles which contain surfactants, and the surfactants prevent the collapse of the lung tissue due to hydrogen bonding when we exhale. And the other features is that the There's also fluid in here which accelerates the diffusion of the gases as well. Notice how tiny the capillaries are, and if you look very carefully at this image, you'll see the tiny 5 to 7 micrometer structures of red blood cells as well. So surface area to volume issues are resolved by changes in shape and differentiating and specializing, folding in, folding out, and flattening. The link to the next video is shown, to the channel, and of course to the playlist.